but I'm Daniel Claborne. I'm a data scientist here at the lab. Been here for 5 years now. Didn't need to collect my swag for being here for 5 years, I think. But yeah, so I started here as a postmaster's. I got my master's in statistics. And when I started, I was doing mostly software tool development and kind of one-off analysis of omics data, all this stuff that they're collecting from mass spec instruments at the lab. So that was usually software tools like you've used, I think, in previous days, as well as applications in case you don't want to do the code and just want to use a web app. I've been involved in some of those as well for mass spec data. More recently, I've been doing investigations into methods for multi-omics as we get some of that data. And so here I am talking about multi-omics. The other half of my work is doing more, I will say, deep learning focused analyses for national security projects. Promise I won't talk too much about deep learning since you got quite a handful of that in the previous presentations, but it's a little hard to avoid. Deep learning finds everyone, including me, eventually. So, okay. Just to motivate this talk, this is about multi-omics. Just talk about, well, obviously, what is multi-omics and why is it important? Some of the methods for extracting insight from multi-omics data. And I think probably most importantly, or at least the thing I want to get across, and I'm glad it was sort of a theme in this series, was what motivates the choice of a particular method. So modeling decisions. Like mentioned previously, you really need to understand what's going on in your data when you select a method and say, okay, is this method going to capture what I care about? Is it going to answer the question that I care about? So yeah, let's get into it. Okay, so just really quickly, what is multi-omics? Oh, there we go. What it sounds like, it's multiple omics. So you have some biological unit in this picture from this paper on multi-omics factor analysis that I'll briefly explain later. They had patients. They measured various things about them. So they have genomics, proteomics, metabolomics measurements on these subjects. And yeah, so you instead of one of these, you have 3 of them. I mentioned some of the things that these different measurements could be, but in the literature, people usually talk about different views. So you're getting a different view of the biological system when you look at proteins versus metabolites, for example. Some of the different views can get a little bit more wild. So when we did a little bit of review of methods, people were trying to inject information from like clinical data. Some people tried to simultaneously combine information from omics measurements as well as like medical scans. I'm going to focus on mostly the omics portion of this, but just to say that these views can be completely arbitrary. Any information you can collect about some biological system or whatever it is you're looking at, people have tried with various success to develop methods to combine all that information. But today we're going to be talking about combining the information from omics data. So in my case, we usually look at proteomics, metabolomics, lipidomics. Okay. So, okay, we have all these views. What exactly do we want to do with them? I'm going to use some big air quotes here. I think I was looking through this the other day and I'm like, I use quotes a lot in this presentation. I don't know if that's a good or a bad thing, but we're going to try to analyze them simultaneously. And a lot of the rest of this presentation is basically explaining what does it mean when I'm saying we're analyzing them simultaneously. And then why would we want to do this? Well, basically, we get a more complete view of the biological system when we include all these different omics. So proteomics is not enough to describe what is actually happening in some biological system. And so more importantly, we're interested in the interactions between these different stages or levels of the omics hierarchy. So yeah, that second bullet point is more about and it's something that I hope I get across in this presentation, being able to do some sort of explainability, some sort of understanding at a kind of like subject matter expert level of what is going on in the biological system. That's something that's very difficult, but you'll see people have developed some methods to try to help someone with subject matter expertise to look at results and say, okay, I know what's going on here, or at least I have like a hypothesis about what's going on here. Kind of separate is that we can just build more powerful predictors. This gets a little bit away from the, we discussed briefly like explainability and deep learning, and I'll show some deep learning methods in this presentation, but sometimes you just care, look, I just want to be able to differentiate between this cancer type and this cancer type, and I don't care how you do it. Give me a system that does that well, and I can do it better if you give me proteomics, genomics, lipidomics than just one of those individuals. All right, so this is a relatively new field with people, these new methods coming out for processing this as the data becomes more abundant. Like we've seen in images, it's very easy to collect tens of thousands, not very easy, sorry. It's easier to collect like tens of thousands, sometimes millions of images. We're not in that domain. So some problems that come up with multi-omics is that we're in this, I don't know if this terminology has been thrown around, but P much greater than N situation, which is to say that usually the number of features we have is larger, sometimes much larger than the number of samples we have. We might have 1,000 features and 60 samples. And basically, when you have a lot of features, it's saying, okay, your system is very complex to describe. In that case, you're going to need more samples to adequately cover the complexity of that system. Some methods for dealing with this you've probably already heard of in previous days, stuff like dimensionality reduction, feature selection. You have 1,000 features, maybe you are able to select some small subset of them that still allow you to do machine learning or statistical analysis. When you have these multiple data sets, they can be hard to combine. It's not just like you have, all your data is just a bunch of normally distributed magic fairyland stuff. Some of them are, you have continuous versus count data, and it's not always straightforward to just combine those in one analysis. Missing values are a problem in omics. As I learned when I got here, people told me, it's like, yes, we have this data set for you. 40% of the values are missing. I go, oh, well, that's a problem, isn't it? They go, yes, we need to deal with that. Here, there's another even worse, another layer of this problem is that an entire view may be missing. So you took measurements on a bunch of patients. Some of them, for some reason, didn't get clinical data measured on them. You don't have the metabolomics for that patient. What can you do in that situation? It's complicated. The last one is the nature relationship between these data sets. It's difficult to model, not obvious, and that's again going back to this point where you need someone to say with subject matter expertise about what's going on in these biological systems to explain to someone who understands the algorithm to choose an appropriate method. Thing works so well. All right, there's the question of, okay, we have all these things, we want to analyze them simultaneously. There are various different ways to do that, and it's probably useful to categorize them into try to categorize them in order to understand how people are going about designing these methods. So like I said, we did a review of some of these methods not too long ago, and in the literature they have various classifications, but I think my main takeaway from this is that while you can do these classifications and it's useful for a presentation like this, really it's about, okay, look at your problem and make the right modeling choice. So, but for structuring a presentation and maybe doing a review, it's good. So I will be talking about it in terms of these three categories: early, middle, and late. Also mixed in with other categorizations in the literature, such as mixed, intermediate, and hierarchical. You can take a guess at what some of those mean, but hopefully I cover them well enough in this presentation here. So like I said, the point of all these schemes is to force the algorithm to learn in a certain way that reflects what's going on in your data. So to do an analogy to the previous presentations, glad how well this flowed together. We didn't plan this. Convolutional neural networks, you say, okay, I am processing images, and I want to know if there's a dog or a cat in there. If I shift the image down a little bit, should that change my answer? Someone who, all of us really, you don't need an expert to tell you that that shouldn't change whether you say that's a cat or a dog. Convolutional neural networks have specific things built into their architecture that says, yeah, ignore that. We should not care about these shifts of the image as long as there's still a dog in there. All right, so we'll start with early integration. Early, middle, late are the categories I'm going to cover here. Throughout this, I'm going to use these, try this laser pointer here. Hopefully this is clear. 3 data sets, these grids representing the rows being your features, right-handed, the columns being your samples. For, pick whatever you want. Let's say blue, red, green is proteomics, lipidomics, metabolomics. So early integration is basically the most straightforward method of doing simultaneous analysis of your multi-omics data, which is to say that, hey, we have the same number of samples, right? For each of these different views. We can concatenate them into some big data frame. And yeah, that is amenable to running through some machine learning algorithm to give you answers, whatever, cancer versus not cancer, disease versus not. So yeah, that's a great advantage in that it's very simple, and maybe it's useful to start as just a quick analysis, exploratory analysis. But as you might guess, there are some problems with this method. So imbalance or heterogeneity across your views may cause it to pay too much attention to one of them, right? And so, like I said before, there may be data-specific distributions like continuous or like count data in one of them, have some continuous normally distributed data. I guess maybe going back to this one, your machine learning algorithms have a tendency to take a look at your big concatenated data frame here and say, oh, I'm just going to do whatever I can. It doesn't matter to do this task well. And so you've said, oh, the blue, green, and red things, the proteomics, lipidomics, metabolomics, these are all important in determining the outcomes that we see. Machine learning model says, yeah, that's great. I can do all that with just the red. And that's not what we want. So like I said, the missingness of a particular view is problematic. Specifically here, you can imagine if one of these, in one of these columns here, suppose you just had an entire block, entire column in the blue of just missing data. That's not easy to deal with. And the high dimensionality problem, like I mentioned, is exacerbated. So started out with stuff that maybe had 100 each rows. You now have a data frame with 300 features, perhaps only 60 or so. Okay, so probably we want to do better. There's another class of algorithm that does this middle integration, and I've thrown in here because I think the distinction between these is a little bit subtle. You see these intermediate and mixed algorithms as well. Generally, as you might expect from the name, they try to combine the inputs at some middle point. And I'll go over a couple examples of what that middle point might be and how it might do it. Yeah. Okay. So here's one scheme. You take each of your data sets and you just process them through separate pipelines to form some intermediate representation and then combine them at some later point. So this has some good advantages that takes care of some of the problems I discussed earlier. It can, as that part of that intermediate step, it can take care of your high dimensionality problem. Maybe it squishes it down to a lower dimensional space, which is more tractable. And it'll remove some of those view specific differences in your data as part of that intermediate step. So some ways people do this. I'll go into examples and explain some of these later, but kernels, essentially intermediate representations about this kind of high level, like the similarity between your observations. If you can represent it, instead of the raw values, represented as some sort of similarity metric between your observations, that's can be better. Graphs are very nice. Maybe you've probably seen these represented by nodes and edges. I'll show an example in the next slide, I think. And like I said, we can't get away from it. People are doing all sorts of stuff with deep learning, and as you'll see, the architecture of deep learning is particularly suited to this process through separate models and then combine at some later point. Yes, okay. So here's an example of some. So it's like, okay, we have an intermediate representation. How do we want to represent it in this middle step? So this particular paper or method called NetDX uses a graph representation. You can see, I showed this before, they have this multi-omics plus clinical data coming in. And they say, okay, for each of these features from clinical data, gene expression, metabolomics, mutations, we're going to form graphs using those features, or we're going to form connections between patients using those features. And then they ask, okay, for say gene expression, is gene expression or a particular gene useful in connecting patients, these blue patients, say, who are the low risk patients, to each other? And is it also useful in connecting the high risk patients to each other, and also making sure that the connections between those two classes are they're connected well to each other within classes and not to the other class? And they do some very complicated scoring method for each time they build these graphs for each feature, and then they sort of score them based on their ability to do this matching of patients within the same group. So they've done a level of feature selection, essentially, to reduce the dimensionality of their data. And then they use those selected features to compare some new observations to known observations. Say, okay, with these features I've selected, are the features of some patient I'm interested in close to patients that we know their risk status? Oh, cool, yeah. So intermediate representation using graphs. So then I want to get into just a discussion about, for intermediate representations, just give some intuition about modeling decisions. There is this question where you're processing them through of your, whatever you want, blue, red, green data sets, proteomics, lipidomics, metabolomics, say, process them through some separate black box method. And you say, okay, each of these could also be used to describe individually some of your outcomes. As opposed to what I kind of described at the start of this about what middle integration does, which is to process them, but then also just combine them so you get like the combined representation of your multi-omics, and then use that to answer your question. But it might be useful to do both. And one of the papers that we looked at, one of the methods that we looked at, I'll explain this in a second, don't worry, addresses that trade-off. So if you stare at this diagram closely enough, try to focus on this part where you have this one input coming in and another input coming in. And like I said, this method also dealt with the situation where, oh, we're actually completely missing one of the inputs for a particular subject. They say, okay, for X1, we're going to form some individual representation for just that data. We're also going to do it for this data, and then we're going to combine them. So this is all maybe a little bit too much detail, but it combines those different views through what they call this product of experts. And this product of experts can handle the missing views by just saying, okay, you only gave me 2. I can still form some sort of representation of that. And in the training data, it just learns to see enough examples where some of the views are missing that it is generally robust against the view missing problem. Okay, and then this last point, which kind of ties together what I was saying with the individual representations versus the combined representations. They say, okay, in this case, I believe they were predicting like survival rates for various patients using the Cancer Genome Atlas data set. Say, okay, that's our task, predict survival rates of these patients. They found that just combining these things together and then using that, or say this combined representation to do prediction was not very robust. And so they said, okay, we're going to actually require that the intermediate representation, just using, say, proteomics, should also be good at predicting survival rates. And so when they train the model, they say, you have to solve the task of predicting survival rates using the global or combined representation, as well as the marginal representations. Okay, so yeah, to review, that was a plan of process everything separately and then combine them. And then there's this other kind of well-represented class of algorithms that makes this, that does this sort of, again, in the literature, this sort of mixed integration. Now, you might look at this and say, this looks awfully similar, but hopefully I get across the differences between the previous thing I talked about. So the assumption here is that you have your multiple views, and what's actually happening is they all exist in this like common latent space, or this common hidden representation of the data, which is actually driving what we're seeing. And this is part of a large class of algorithms there are people out there who have spent their entire careers doing like very sophisticated matrix factorization methods. They're all generally based on that kind of stuff. Some advantages: don't need prior transformation to learn this shared representation. So your data comes in, we don't need to tell the model much to learn this hidden representation. But it's a little bit more sensitive to heterogeneity in the data. Okay, here's a picture of basically what's happening. You might have noticed I changed the sizes of the boxes here. That's intentional, but we say, okay, we have these 3 data sets. What's really happening is they exist here in this latent space, and each of these data sets actually has this little weight matrix that is mapping from the latent space to the what we actually observe in the data. So again, you say, okay, we have these 3. This is actually just a bunch of weight matrices that are taking values from this hidden latent space, from this latent space. Okay, and I I guess I just want to mention that, yeah, we have the same number of samples, and then I have these 3 different sizes of number of features for these data. Okay, so some useful questions for some of the popular methods that are out there. I think these are actually things that can be used. A popular method called multi-omics factor analysis that I showed a picture of before. You might want to answer questions like, okay, how much variation does this latent space, this hidden space, explain in a particular view? How much does it explain about my proteomics? How much does it explain about my metabolomics? And you might also be interested in how much does a specific factor explain about each of those. So one of the nice things about latent space methods, these matrix factorization methods, is you look at the factors, and someone with domain knowledge can take a guess about, okay, this is the factor that is driving variation in, say, proteomics. It weights heavily on certain features that I know something about, and you can use your domain knowledge to say, what about the biological system is that factor capturing? Yes, okay, and oh yeah, and you might find factors that actually drive the variation across multiple views. So those ones might be of particular interest because those are the things that are, maybe we'll call them the multi-omics factors, things that sort of link all of the different omics together. They describe variation in all of your omics. Okay, I always go over some of the issues for these. Like I said, a little bit sensitive to the differences in the views. And is this assumption they've made about all your data lives in this weird hidden representation, is that a reasonable assumption? Again, that's something for domain experts to decide before they start doing these analyses. So I'll describe a method that we're actually going to look at and play around with later today in the hands-on sessions called sparse partial least squares. Here's a picture of it. It looks probably very similar, but with a subtle difference. So just 2 data sets now. It has the same thing where we say, okay, this data set is actually some weight matrix times this hidden latent representation. But you'll notice here there are actually 2 latent representations, not one shared one. But the way this algorithm works is it says, okay, represent these 2 things that I observe this way, but I also want you to make these 2 things, similar. So the latent space for your proteomics, lipidomics, tell the algorithm, hey, do your best to represent the lipidomics this way and the proteomics this way, also under the constraint that their hidden representations should be somehow similar. In this case, it's high covariance. Yep, cool. Some plots. People like to look at plots, and you will get to see these plots later. We'll make these plots, and I cannot hover over them on the presentation, but you would be able to see that this is, I think, a lipid, and these yellow ones are a protein. So you want to look for common sources. Like I said, you might want to look for common sources of variation in those latent factors. I'm trying to think to explain this. Yeah, this circle plot, I'll try to do this as best I can, captures how much, say, this lipid correlates with one of your latent factors. So here I'm just plotting 2 of the latent factors. It says, okay, it is highly correlated with this one. So it's all the way over here at high correlation with this latent factor represented by the x-axis, and then not so much on the second one. In this data set, there's really not much more, it seems. The blue ones, they're all over the place in the latent space. But something else useful you can do is say something about, take a look at the lipids and proteins that lie in the same region in the latent space. So perhaps these ones are of interest, and you can go and look at what those proteins and lipids are and say, aha, what is going on here? Probably seen this graph before, loadings. So for a particular component or a hidden factor, what biomolecules is it sort of picking up on? What parts of the underlying biological system does it capture? And so you would look at this and say, okay, these orange ones weight heavily on the first component. Yes, okay, I was making sure that the first component, and that first component this is how it's represented. These are the biomolecules that are heavily represented in that component. Okay, oh yes, we're going to late integration. Late integration, so the middle integration stuff is usually the most complicated stuff. Late integration is the other simple thing you could do, which is to say, do this thing where you process them through separate networks, but we're not really going to combine them. We're just going to say, okay, everybody do what you can, best can by themselves, and we're just going to combine your output at the end. So this usually takes the form of, who voted that this was cancer type A. You say, okay, most people voted it was cancer type A. We'll say it's cancer type A. This loses some of the explainability that you might get from intermediate methods. By yes, sorry. Okay, sorry. Some of the advantages first. So yeah, we run them through, aggregate them at the end. So yeah, advantages to these, obviously, it's easy. You can run them through whatever model you want, and then just combine prediction results at the end. It possibly, maybe counter to intuition, it might actually do the best. I've looked at systems where they do this fancy thing in the middle where it's going to learn the interactions between them. But it turns out for just a prediction task, the best thing you can do is just to average the outputs. And that's actually better than doing all this fancy stuff. But we lose a little bit of this intuition about the relationships between our different inputs. So it doesn't learn these relationships. We can't do anything there. And yeah, generally it's kind of unpalatable from an explainability standpoint, especially if this is some neural network thing. No idea what's going on here. That's why I presented them my black boxes, and then we just aggregated their votes. Not much you can do there from an explainability. Yes. Okay. I guess I don't have an example for this one, but I just wanted to give this as an example of maybe some of the more biologically informed ways people do multi-omics integration. So the ones I showed, they sort of process them in parallel in a sense, right? Everyone just gets a little thing. They go through their own little model. Maybe we combine them. But as some subject matter experts might tell you, that's not really how things are happening at the biological level. You remember, I said I came out of a stats major, not a biology major. So one of them might tell me, that's not how the interaction between genes and proteins works. It's more of this hierarchical thing. So you might want to say your model actually starts with something, say at the gene level, then we go to the protein level, and then you do your outputs. Whatever you want to imagine green is here. So yeah, modeling considerations. I'm not familiar with an actual method. I think there were some graph-based methods for this type of hierarchical thing. They're very good at that because you can say from this node to this node, only allow a relationship in one direction. That's one way people have designed models that follow this sort of hierarchical architecture. Okay, so conclusion, multi-omics, right? It's one omics type is not enough. We need to have a complete view of the biological system to really understand what's going on. There's really big diversity in the number of ways you can analyze multi-omics. So, one when I started looking into this, I'm kind of experimenting with methods like the couple I showed you before and developing these tools. But when I looked into it, I was like, okay, there's these categorizations of these methods, but really it's just a bunch of bespoke things that seem to have some commonalities. For imaging data, there are some choices you have to make there depending on what the problem is. But for images, for a long time, this is, okay, you're doing images. Okay, it's going to be some kind of convolutional neural. My sense is that it's not the case in multi-omics. It's not the case that someone says, here's some multi-omics, and you say, oh, you're going to need to do this like blockwise matrix decomposition on this. That's not the case. It really depends on the specifics of the study. It's incredibly complex. And like I said, yeah, basically what I just said. You have to be very careful about what model you choose to analyze the multi-omics data sets, because like I mentioned before, they have a tendency to give you spurious conclusions. It says these things were important in determining the outcome, but really it's just what the machine learning model decided was the path of least resistance to get you to your answer.